you can't hear me, just let me know. So, hello, good afternoon. Thank you so much for coming and sticking around for the last talk of this session. My name is Clara, and I will be talking to you about what can lichens tell us about the latitudinal diversity gradient. So we're switching gears a little bit from diversification to just thinking more about general patterns of diversity. So as always, there are many, many people to thank. This has been a very large-scale dissertation project, and I certainly could not have accomplished it without many, many generous, patient, and brilliant people, and also various funding sources as well. So just a really quick outline, I will introduce you to lichens and the latitudinal diversity gradient, then get very briefly into my methods, which I'm happy to talk more about later, some patterns that I found, and some potential drivers, and then conclusions and some future directions. So many of you, if you think about lichens at all, might picture something like this. These are in the genus Cladonia, and their common name is pixie cup lichens. But I, as a lichenologist, will not dare to say what species these are because they're actually quite difficult to distinguish. Some of you may be more familiar with another Cladonia species, which has little stalks with little red caps on it and is commonly known as the British soldier lichen. So you can find these growing on the forest floor or on old stumps in the forest and things of that nature. And they tend to be the sort of lichens that people picture. But lichens come in all shapes and sizes. This is also a picture of lichens. In fact, everything in this photograph, except for this tiny little bryophyte sticking out over here, is a lichen. So this is a photograph of the side of a tropical tree, but nothing of what you're looking at in this picture is actually bark. It's all different species of lichens. And these are what we refer to as a crustose growth. Lichens have been defined as a symbiotic relationship between fungi and a photosynthetic partner, either algae or cyanobacteria, or sometimes both. And so, of course, these are two different little cartoon depictions of lichens. So over here, we have, let me just see if this other piece works better. Without like shining anyone in the eyes. Yeah, okay, so over here we see a nice little cartoon of a fungus and an algae making friends. And then over here, what we have is a cartoon version of if we took a cross-section slice through a lichen. So the blue is representing fungal hyphae all along here, creating most of the structure of the lichen that we see. And then we have this layer of algal cells. And the symbiotic relationship, of course, is that the algae perform photosynthesis and produce sugars in exchange for basically shelter, water, and nutrients from the fungus. And one thing I want to point out here, so that I don't forget to say it later, is that because the fungus is kind of the dominant part of the structure of the lichen, when we talk about a lichen species, that's what we're referring to as the fungal partner. One of the reasons that I love studying lichens is they like to break all the rules. As soon as we think we know everything there is to know, there's so much more that we discover. And so these are just two pictures referring to two different articles about finding even more symbionts potentially involved in the lichen symbiosis. And so this simple one-to-one relationship is never really that simple. And I really enjoy this title by Ed Yong, Lichens are the overlooked organisms that keep challenging our assumptions about life. And so again, that's why I really love studying them and why I thought they would be a fantastic system for looking into the latitudinal diversity gradient. So what is the latitudinal diversity gradient in all of these maps? Warmer colors refer to higher species richness, and then cooler colors refer to lower species richness. And so in each of these maps, which include mammals, birds, amphibians, and vascular plants, we see this pattern where we have really high species richness in the tropics around the equator, and then as we go either north or south with increasing latitude, we see decreasing species richness. So that's the latitudinal diversity gradient, and it's not just a terrestrial pattern, it's also observed in many marine taxa as well. This pattern has been observed since basically the time of the early explorers. Humboldt, Wallace, and Darwin all commented on the fact that they saw way more diversity, basically life upon life in the tropics, and then less as you go towards either of the poles. And again, based on these maps and many, many studies, this has been demonstrated in many, many different groups of organisms but not lichens, at least not at a very large scale. So that is what I set out to do. I'm not going to get into too much of the hypotheses behind the latitudinal diversity gradient. So basically, for as many times as scientists have looked at the latitudinal diversity gradient, they've come up with new and different hypotheses of why it exists. And to date, there's no real consensus on a single hypothesis. But of course, as with most of biology, it's likely a combination of factors, evolutionary and ecological. And what I'm just showing here is that, so we have this latitudinal gradient of species diversity, but by the very nature of the roundness of our planet, the distance that it is from our closest star, the sun, we also have this latitudinal gradient of mean annual temperature and angle of incoming light. And so regardless of what other factors may be involved, we always find these correlated with any latitudinal gradient patterns that we find. So just getting into the methods, so I love lichens, they're really interesting. There's this really big global pattern that's been demonstrated for a lot of taxa, but not for lichens. So let's go forth and find out what's the dealio with those. So I visited 9 different field sites. These are all various long-term research or long-term protected forest sites. So in an effort to narrow down the scope of my project, I only focused on lichens that grow on the bark of living trees, so epiphytic lichens, and only on the lower parts of the trunks, basically researcher height. So that was my sampling thing. So I targeted forest sites basically along as broad a gradient as I could, from tropical forest regions up into the boreal forest in Canada. And yeah, so a lot of these, I won't get too much into this for this talk, but happy to talk afterwards. A lot of these sites had tree data already available to them, which enabled me to have a more random sampling approach, but also enabled me to look at the impact of host tree on the lichen species richness that I found. So again, this is just a little schematic breaking down what I did at each site. So at each of these 9 sites, I randomly chose sort of 10 plots within the forest. At each of those 10 plots, I randomly chose a tree and then its 9 closest neighbors, totaling 100 individual trees sampled at each of these sites. So 900 trees sampled across this gradient. And again, I was sampling within a fixed area on each tree at researcher height. So what did I find? We're just going to jump right in to this. So I have p-values and r-squared values, but I basically have 9 data points in everything that I'm showing to you, so do take these with a grain of salt. And I'd be very happy to talk to people afterwards, especially after this morning's talk, about the best approach to looking at these data in a meaningful way. But if you just look at the data here, so we have latitude on the x-axis, starting at my southernmost Peruvian Amazon site at 12 degrees south, and then we have lichen species richness. And we do see generally higher lichen species richness in the tropics and around the equator, and then not quite a steady decline, but declining species richness as you go north, and certainly the lowest at the northernmost boreal site. So my conclusion from this, and from another chapter I worked on looking at digitized herbarium specimen data, is that lichenized fungi do actually follow a latitudinal gradient. So we can add those to the large and growing library of taxa that follow this global pattern. Interestingly enough, I know these plots are a little bit hard to see, but I just wanted to add them as a visual. So this is looking at beta diversity. The top one is a measure of similarity as you go from one plot to the next within each site. And then the 2 bottom ones are different metrics of dissimilarity. And so what we see, so this is again my most tropical site here, and then all the way up into the northern boreal. So as latitude increases, similarity increases and dissimilarity decreases. So we have, as we move from one plot to the next, we find new lichen species. Whereas here, I was basically finding the same lichen species on each tree that I sampled from. So again, that has been shown in other taxa as well, this pattern of beta diversity along the latitudinal gradient as well. So I was interested in looking at some functional diversity in lichens in addition. This is way more lichen biology than you will ever need to know, probably, but I'm just breaking it down into different growth form types. So I mentioned really rapidly at the beginning about crusts being those really two-dimensional structures. We, of course, have very creatively come up with many other types of growth forms, but I'll summarize them in this next slide here, just in terms of more two-dimensional growth forms versus more three-dimensional. And that's shown in these pictures too. This is a more crust-like growth form here, very oppressed to the substrate. And here's a really beautiful foliose. This is Loberia pulmonaria, which again, if you've heard of lichens at all, you may have heard of that one, the lung lichen. So this is much more three-dimensional in structure. It's actually like, curling off of the tree here with gorgeous sunlight shining through. And these are also interesting in that their thallus itself, so the lichen, has a much more organized structure to it in these three-dimensional growth forms than in the two-dimensional ones. What we find is a significant shift along the latitudinal gradient from much more two-dimensional growth forms to much more three-dimensional at the same height on trees as we go along this latitudinal gradient. And I'll get into a few reasons why I think that might be in the next section. I was also interested in looking at another functional trait, which is reproductive mode. So some lichens reproduce sexually, which again, because we think of lichen species in terms of the fungal partner, this is the fungal partner sending off spores that have to land somewhere and find a new algal partner to associate with. They can also reproduce in a variety of asexual ways, but this includes basically just sending off fragments of fungal hyphae and algae to go off and create a new lichen cell. And so I wanted to look at the ratio of asexual to sexual reproduction along the latitudinal gradient as well, but we don't really find a pattern there. This was a bit surprising to me at first because some studies have found a gradient as you move from one habitat type to another in terms of ratio of reproductive mode, but even though we're traveling across very different forest habitat types, these are nevertheless all forests, and so maybe this isn't as surprising as I think it is, but again, maybe this needs to be probed into further. So this is just an image showing these are some apothecia, which are fungal structures that will send off spores. So that's an example of sexual reproduction. And then right up here, we have a lichen that has ceridia on it, which is those little fragments, so asexual. So this is going to be very brief, and again, you'll see from my models that I think we need to use some better models here, but I just wanted to look at some potential climatic drivers of these patterns. So in terms of lichen species richness, we broke these models down into three sort of different categories, basically annual means and what impact those have, seasonality of temperature and precipitation, and then actually looking at extremes of temperature and precipitation and which one is the biggest driver. This, again, according to a ludicrously high R-squared value, was our best model. But in terms of thinking about lichen biology and the fact that many lichens are in fact extremophiles, it makes sense to me that it is the extremes that may be driving patterns of diversity at this global scale. In terms of the growth form, so I thought I would come back to this. Again, in general, the best models is extremes, but in both the seasonality and the extremes models, the precipitation variables that are significant. And one of the things about the lichen growth forms is the more three-dimensional structures would be better at getting moisture out of the air, whereas the more two-dimensional might be better at dealing with saturation. And so that may, again, make sense as to why we see that along this latitudinal gradient from really moist tropical forests up into that northernmost boreal site is actually quite a dry boreal site. Finally, some conclusions of future directions, because I think I only have like 30 seconds left anyway. So just to wrap up what I told you, so lichen richness and beta diversity do seem to follow a latitudinal gradient based on these 9 sites. Lichen richness is driven by extremes in temperature and precipitation, and lichen growth form follows a significant shift along the latitudinal gradient and seems to be mainly driven by precipitation patterns. We looked a little bit at lineage-specific patterns, but unfortunately we had too little data to find any significance in those, but basically different lineages of lichenous fungi do follow different patterns along the latitudinal gradient. And my next chapter, so stay tuned for the next conference, is looking at some possible diversification in actually the different partners of the lichens. So thank you very much. I think we're out of time, so I don't want to keep people from posters, but you're welcome to come up and ask me questions if you want.